Movies about the future often feature artificial intelligence, aliens from other planets, armored suits for flying around, and replicators that can make anything you want from scratch. Human beings in these movies, though, don't seem very different from us. That our own biology may be altered in ways that do not involve fusing with flies or being bitten by radioactive spiders or other comic book superpowers is rarely reflected in these visions of the future. What's missing is biotechnology. The term biotechnology very loosely refers to any combination of biology and technology. In that sense, the first humans to make bread or beer or cheese were applying their knowledge of biology to make things. But what modern biotechnology usually refers to is the more specific application of biomolecular knowledge about the underpinnings of life. Molecular biology has given scientists and engineers the ability to manipulate the recipes written in our genes in ways that could not even have been imagined in 1953 when the structure of DNA was determined. In this lecture, we'll trace a few of these remarkable developments in biotechnology. You'll learn about advances involving reprogramming cells. We'll talk about stem cells, the cellular building blocks from which everything in our bodies is sculpted, and how these can be used in medicine. We'll talk about a technique that can be used to make clones of beloved pets, endangered species, and perhaps bring back an extinct species like the woolly mammoth. And we'll explore advances in creating stem cells from adult non-stem cells and the many options that such cells open up. Then we'll move on to initiatives in the field of synthetic biology that allow us to create genomes that have never before existed and use them to program cells, essentially creating new life forms. There's a lot to cover here. Let's go. Biotechnology in its current form began in the early 1970s, when scientists figured out that DNA from organisms as diverse as elephants and amoebas had the same structure and uses the same genetic code. As a result, one can insert an elephant gene into amoeba DNA, and seamlessly, the cut and pasted gene becomes a part of the DNA it's inserted into. The cut and paste metaphor works because DNA molecules can be cut using highly specific enzymes and joined to other DNAs using DNA ligase. Because this combines DNAs from two different sources, it is said to be recombinant DNA. Now, not only that, if the elephant genes are inserted so that it is near a promoter that can regulate its expression, the gene can be transcribed, processed, and expressed in the amoeba, just like the amoeba's own genes. In other words, a recipe from the elephant cookbook can be integrated into the amoeba cookbook and can be used by the amoeba. It's basically using the amoeba's RNA and protein synthesis machinery to build a protein to elephant specs. The protein made in this way is called a recombinant protein. Now, why would anybody want to do this, you might wonder? No, it's not because anybody wants to make an elepheba or an amoebafent. Researchers developed the method because it's useful to be able to produce proteins that are otherwise hard to come by. Human proteins, for example, can be made in yeast or bacteria, which serve as factories for building proteins according to the instructions provided by the human genes inserted into these cells. So why not get the proteins from the cells that actually make them? Well, it can be hard to lay your hands on a few kilograms of human brains, for example, for a study on specific brain protein. Making recombinant protein gets around this problem. The same method also allows us to make proteins for medical treatments. The first recombinant protein made for therapeutic purposes was insulin, which was approved for use in 1982. Recombinant insulin under the name Humulin was made by inserting the gene for human insulin into bacteria. The insulin produced in the bacteria is identical to the stuff we make in our bodies because it is made using the same recipe our cells use. Prior to this, patients who needed insulin mostly relied on insulin extracted from pigs. Pig insulin is quite similar to its human counterpart, but it is a foreign protein and can trigger an immune response. Patients were then in a bad place. They couldn't control their diabetes without insulin, but their immune systems would attack the pig insulin. Because recombinant human insulin is indistinguishable from our own insulin, it does not cause these problems. dozens of medical-grade proteins that have been made using recombinant DNA technology, including human growth hormone, clotting factors, and anticoagulant proteins. But where the initial techniques used bacteria or other cells grown in culture, some of these proteins are now made in animals. For example, fertilized eggs of goats can have a gene for human growth hormone inserted in them and integrated into their chromosomes. The genetically modified goat embryos are then implanted into a female goat. Goat offspring obtained in this way are transgenic. They contain genes from an alternative source. Most importantly, they make human growth hormone and secrete it into their milk. It's cheaper and easier to take care of goats than to maintain laboratory cultures of cells, and the goats keep producing the milk over a long period of time. Also, once a transgenic goat has been made, it is relatively easy to create a whole flock of animals that can produce the desired protein. And what do we call making pharmaceuticals in animals? Farming. P-H-A-R-M-I-N-G, of course. Recombinant DNA technology revolutionized biomedical research. It also made it possible to improve important crop plants. One of the biggest success stories in agriculture was the production of a virus-resistant papaya that saved the papaya industry in Hawaii. In the early 1990s, papaya growers were hit hard when the ring spot virus devastated plantations. Researchers reasoned that if they could insert a gene from the virus into the papaya, the plants might be able to fight off an infection. Although plants don't have an immune system, there had been reports that exposure of plants to a virus could help them resist a future attack. We'll see why in a minute. So a gene from the virus was shot into the papaya plants using, I kid you not, a gene gun. A gene gun is a device that literally shoots DNA-coated particles into cells. It's especially useful for getting DNA into plant cells, which have a cell wall, not just a cell membrane around them. The genetically modified papayas made this way were resistant to the virus. We now know that introducing a virus gene into the papayas was successful in preventing virus infection because of RNA silencing that we've talked about. As you've learned, an RNA copied from the inserted virus gene can seek out and bind to RNA made by the same virus if the virus infects again. The inserted gene can block translation of the infectious virus RNA or even target it for destruction. Either way, the virus itself is stopped in its tracks. Insertion of the virus gene into papaya saved a multi-million-dollar industry. Transferring genes into cells is one very important method in biotechnology. Another one arises from the biology itself, and here I'm referring to stem cells. There are two things that are special about stem cells. One is that they are capable of dividing indefinitely, that is, as long as the organism is alive. The other is that they are undifferentiated. They're like a child who hasn't yet chosen whether to be an astronaut, a ballerina, a surgeon, or an artist. When a stem cell divides, the new cells may differentiate, or they may remain as part of the stock of stem cells. In the earliest stages of embryonic development, the fertilized egg must divide to produce a large number of unspecialized cells before those cells are assigned a specific function. These embryonic stem cells, or ES cells, undergo specialization over time, turning into all the different cell types we have in our body depending on the signals that they receive. This means that if we know what signals to provide, we can use stem cells to create new cells to replace cells that are injured or lost due to disease or trauma. For example, they could be used to replace neurons in people with spinal cord injuries or provide new heart cells to people who have suffered damage to their heart muscles following a heart attack. Because of ethical concerns surrounding the use of cells obtained from a human embryo, alternative strategies have been explored. One solution is the production of what are called induced pluripotent stem cells, or iPS cells. Pluripotent refers to the ability of a stem cell to give rise to many other differentiated cell types. To do this and yet avoid working with cells from a human embryo, scientists begin with differentiated somatic cells. Now, these are cells from the skin of an adult, for example. Scientists must then reverse the differentiated state of the cells in the laboratory to return them to the condition in which they are once again capable of becoming any type of cell. By comparing embryonic stem cells to differentiated cells, researchers were able to identify a handful of genes that were expressed at high levels only in embryonic stem cells. When scientists increased the expression of these genes in differentiated cells, the cells reverted to a state similar to that of the undifferentiated embryonic stem cells. That is, once created, these induced pluripotent stem cells can, if given the right signals, differentiate into a variety of different tissues. These so-called iPSCs have been used in labs to produce beating heart cells, motor neurons, light-sensing photoreceptor cells, insulin-producing pancreatic cells, even egg and sperm precursor cells. All that has been necessary is providing the right developmental signals to the iPSCs. Muscle cells produced from these stem cells have been tested in a mouse model of Duchenne muscular dystrophy. In 2017, Japanese researchers reported that monkeys with Parkinson's showed great improvement after treatment with dopamine-producing neurons derived from induced pluripotent stem cells. In 2018, clinical trials with humans were begun using iPSCs to treat Parkinson's, heart disease, and macular degeneration. A related technique, somatic cell nuclear transfer, is sometimes called therapeutic cloning. The aim here is not just producing undifferentiated stem cells, but also an entire organism. As the name suggests, this method uses the nucleus from a somatic cell, that is, one that is not a reproductive cell. The nucleus of the somatic cell is extracted for placement inside an unfertilized egg, which has had its own nucleus removed. The inserted nucleus then provides the genetic material for the unfertilized egg, known as an oocyte, which is induced to develop as if it were a fertilized egg. Now, the idea here is that the somatic cell genes in the oocyte environment will be activated or inactivated as appropriate for a fertilized egg. This will restore the implanted genome to behaving once again like an embryo. Somatic cell nuclear transfer is especially useful for making donor-matched stem cells that can be transplanted back into the patient. Because the DNA in these cells is a therapeutic clone from the patient, the cells would not be seen as foreign by the patient's immune system. Somatic cell nuclear transfer has been used to make entire animal clones. This was the technology that was used to make the famous cloned sheep Dolly in 1996. To make Dolly, scientists used an oocyte from one sheep and placed it into the nucleus from a mammary gland cell of another sheep. The resulting embryo was implanted back into a ewe and allowed to develop. Now, why, you might ask, would someone clone an animal when animals are quite capable of reproducing offspring? One reason is to create an exact replica of a particular animal, perhaps a prize bull or a superior racehorse. It used to be that once a prized animal was past its prime, breeders had no option but to look for another animal and hope for it to be as good as the original. With a clone, there's no need to leave it to chance. For instance, a famous champion polo team from Argentina rides using a stable of 100 cloned horses. In polo, you're allowed to and expected to change horses during a grueling match. But what members of this team can do is change from one genetically identical horse to another. Pretty cool. Cloning in the plant world is common. From common potatoes to the Cavendish banana found in supermarkets all over the world, plant cloning is easy because plants can be completely regrown from a tiny amount of tissue. With somatic cell nuclear transfer, cloning of animals is now possible too. China has announced ambitious goals of cloning up to 1 million cattle. Why? Well, clones provide uniformity of quality, and as the joke says, with a clone, you truly can have your cow and eat it too. In fact, cloned animals can also reproduce just like their conventional counterparts. Whether the ambitious goal in China will be met is not clear, though. The practicality of any cloning project is colored by a success rate of somatic cell nuclear transfer that is very low. In some cases, only 2% succeed, and even a very high success rate might be only about 40%. But some people are willing to pay big time to clone their pets. In 2005, scientists in South Korea successfully cloned a dog they named Snuppy short for Seoul National University Puppy. Though costs are declining over time, the biggest factors are the technology required and the low success rates. Until these change significantly, pet cloning will tend to be limited to the wealthy. Animal cloning might help save endangered species. Scientists have taken nuclei from somatic cells of an endangered animal and inserted into the oocyte of a closely related non-endangered species. An endangered wild cow, for example, was cloned successfully this way and displayed in the San Diego Zoo. There are also the possibility of de-extinction of species that have already died out. And no, I'm not talking Jurassic Park here. Dinosaurs died out 65 million years ago, and so it's unlikely we can recover stable DNA that's more than thousands or even possibly hundreds of thousands of years old. But more recently extinct animals like the dodo or woolly mammoths are excellent possibilities. Mammoth remains have been found frozen and excellently preserved. An elephant surrogate mother might be successfully used to bring a woolly mammoth calf into the world. So we've seen that we can move an existing gene from one cell to another to get the surrogate cells to make gene products we want. We can also reprogram the cells of an organism to do everything from fixing problems in the organism to making an exact copy of the organism. The third topic of biotechnology I wish to talk about relies on genomes, the enormous body of knowledge we have acquired from efforts to determine the DNA sequence of entire organisms, including humans. Let's say that instead of moving a single gene, that we want the output of an entire metabolic pathway. DNA and the genetic code happily supply us with a single protein, such as putting human growth hormone into a goat so that the goat's milk will include the hormone. But when the desired output is not a single protein, we're likely to need an entire metabolic pathway of enzymes to make molecules so that we can collect the molecules. So how do we copy all the enzymes of an entire pathway? Well, first, computer algorithms can analyze genome sequences and help us figure out what enzymes a particular organism makes and what additional ones might be needed to carry out a particular metabolic pathway. For example, scientists have engineered yeast cells to help synthesize an anti-malarial drug called artemisinin. This is a compound that is made in a plant, Artemisia annua, a relative of the wormwood plant from which absinthe is made. Unfortunately, the amount of artemisinin made in these plants is small. Thousands of acres of Artemisia would have to be grown, harvested, and processed to get enough of the drug to meet the need. This would be land-intensive and labor-intensive, and the yield is unreliable. On the other hand, producing artemisinin in the laboratory using organic chemistry is too complicated and too expensive. So researchers decided to use cells, which, as you know, are already capable of carrying out complex chemical transformations. Cells can handle a new reaction as long as they have the necessary enzymes to help out with the difficult part of the synthesis. The strategy was to make the molecule artemisinic acid in yeast cells, then to chemically convert the artemisinic acid to artemisinin using standard chemical synthesis. That last step is easy to do outside of cells. This is a bit like a company outsourcing initial steps in the production of a widget to an outside factory that can do it cheaply and efficiently, and then finishing up the assembly at their own plant. Artemisinin can be made from farnesyl pyrophosphate, or FPP, an intermediate in the same pathway that's used in making cholesterol, carotenoids, and vitamin E. Yeast cells already have the enzymes to carry out the synthesis of FPP, so the researchers cranked up the expression of these enzymes. Then they added in genes from Artemisia needed to convert the farnesyl pyrophosphate made by the yeast to artemisinic acid. Once all the enzymes for the pathway were in place, yeast cells were able to make artemisinic acid in large amounts. That in turn was converted chemically to produce artemisinin efficiently and at a price point that is realistic for the developing countries that most desperately need anti-malarials. Okay, so we've seen that beyond inserting a single gene into cells to produce one protein, biotechnology has enabled entire pathways to be constructed to make molecules we want in cells. the yeast or bacteria where the new pathway is inserted can handle it because, one, they're just following orders given by the coding for the inserted genes, and two, the energy cost of making the new enzymes doesn't break the budget of those cells. So stealing a pathway from a cell is a pretty cool advance. But what if you wanted to create a pathway that doesn't fully exist in one cell, but individual reactions of it might occur in 30 cells? How would you do it? Well, one strategy is to make an entirely new cell by creating a biotechnologist's version of designer genes in the form of designer genomes. This is actually possible thanks to two advances. First, we have knowledge of the contents of thousands of genomes. Second, we have developed very efficient and sophisticated chemistry to chemically synthesize DNAs in the laboratory. We can make any sequence we desire, and best of all, machines do all the work. At first, the chemical process was relatively inefficient, so only short oligonucleotides on the order of 20 to 30 bases could be made. But improvements in technology soon made it possible to cheaply and accurately synthesize not just individual genes, but entire genomes. In 2010, a research group headed by Craig Venter reported that they had created the entire genome of the bacterium Mycoplasma mycoides from the known sequence stored in a computer. They then removed the genome from a bacterial cell, inserted the synthetic genome instead, and showed that the cell now was controlled by the synthetic genome, the first time ever that a cell was running on synthetic instructions. Okay, so what Venter did was simply make the DNA according to the sequence that we already knew for the bacterial genome. But there's nothing that says we can't create genomes that don't already exist. Genomes could be designed for bacteria so that they could make biofuels based on metabolic pathways that don't exist in any single cell. Genomes could be designed for plants to enable them to synthesize brand new drugs instead of fabricating the drugs with much slower and more expensive reactions elsewhere in chemistry. We could even rewrite our own human genome, and there may be some enormous advantages. Some people have proposed doing just that, dubbing the project Human Genome Project Write, to distinguish it from the original project to sequence the human genome, dubbed Human Genome Project Read. So what could writing genomes do for us? Well, let's first of all get away from thinking about this as a way to make human beings. That's something scientists agree needs to await a full discussion of the enormous ethical implications. Personally, I don't ever want to go that route, but that's another matter. What I do support are some fascinating novel aspects of the technology. What I'm talking about is making artificial human cells, not human beings. Growing human cells in cultures, in laboratories, independent of human beings is already commonly done. It's accepted, ethical, and not harmful in any way. We could do the same with artificial human cells we might make. That is what I'd like to focus on here. What might be some advantages of making artificial human cells? Well, how about creating human organs for transplant into real human beings? With the right engineering, such organs could be customized for each recipient, so rejection wouldn't be an issue. What about creating custom stem cells that could be used to regenerate an immune system for a person who had theirs destroyed by radiation therapy to kill a cancer? What about custom making retinal cells to replace ones lost by macular degeneration? What about replacing pancreatic cells responsible for making insulin that are damaged in diabetics? Virtually every technology that has been proposed for stem cells could be accomplished with artificial human cells. There are enormous technical hurdles to overcome to realize Human Genome Project Write, but the many benefits may well justify working to overcome them. In closing, I want to talk about two exciting biotechnology advances that did not arise from research in molecular biology, but that will likely impact human health. The first involves nanotechnologies from non-biological chemistry and electronics. In 2018, researchers at China's National Center for Nanoscience and Technology reported development of nanotech robots that could cut off the blood supply to tumors. And what they did was attach a small folded scaffolding protein to the blood clotting enzyme thrombin and injected the modified enzyme into the bloodstream of a mouse with an endothelial tumor. The system also had attached to it a small DNA molecule that specifically binds to a protein called nucleolin, which is found only on the surface of endothelial tumor cells. When the scaffolding protein binds at the tumor, the protein unfolds, exposing the thrombin enzyme. Thrombin catalyzes activation of the zymogen known as fibrinogen, forming fibrin, and the fibrin polymerizes to form blood clots. Thus, thanks to these nanobots, tumors got surrounded by blood clots, which restricts their access to the blood supply and needed nutrients. Consequently, tumors in the treated animals shrank. 3 of 8 mice receiving the treatment had complete regression of their tumors. Best of all, there appeared to be no side effects, such as unintended clotting or immune system reactions to the injected nanobots. The second advance comes from sharp observations of a more traditional kind. This involves the unusual case of a woman named Joy Milne, who complained of a continuing woody, musky odor from her husband Les. A few years later, after Les was diagnosed with Parkinson's disease, Joy attended a support group with him for sufferers of the disease, and she realized that the sufferers of the disease had the same smell. When tested with only t-shirts from sufferers and non-sufferers of the disease, Joy was able to correctly identify all the sufferers of the disease and misidentified only one non-sufferer. And then, amazingly, the apparent non-sufferer also later developed the disease. As a result of Joy's sensitive sense of smell, researchers are now attempting to identify and detect the odorant molecules apparently being released by Parkinson's patients using a technique called mass spectrometry. In this method, trace amounts of molecules are identified by precise determination of their molecular masses. The hope, of course, is that if the molecules Joy was smelling can be identified, then detection and treatments for the disease can begin much earlier than currently possible. Of all the topics in biochemistry, there is nothing that raises hopes and fears as significantly as biotechnology. Frankenfoods, maybe even Frankenstein's monster. On the other hand, save lives, eliminate human disease, extend life without limit. Applications of biotechnology go far beyond the laboratories employing them as tools for understanding. Perhaps there is no human invention short of atomic weapons for which there is a need for intelligent regulation moving forward. Let's all hope that citizens and our legislative bodies can keep on top of the knowledge required to support and enact meaningful legislation to encourage development while also protecting the public.